Bio 235 students, welcome. This is a video overview as we approach week 7 and our first practical exam in microbiology. The intent of this video is to walk through some of the key information in terms of the structure of the exam and also some preparatory tips as you begin to look at your labs in preparation for the due date this coming Sunday. The lab practical is 50 questions in totality with 50 minutes allocated for the exam time. The exam covers the first 6 weeks of labs. They are not cumulative, so your next lab practical will cover weeks 8 through 12. This particular exam is a combination of multiple choice and very short answer fill-in style questions. The lab material that is assessed usually has to deal with the background information as well as the procedural information used to set up your lab. So what I want to do now is walk through and show you areas that you should be paying close attention to as you go through and prepare and study your labs. Each lab cassette for the weekly labs contains a series of background information. It is really important for you to go in and read and review the background information as many of the lab practical questions come from this information. Specifically, make sure that you are familiar with, for week 1, things like the types of microscopes that microbiologists use in the laboratory, starting with the dissecting and ranging all the way up to the electron microscopes. Oftentimes, you will also need to know the labs that you did during the particular week. For experiment 1, I will not ask you questions on the virtual microscope. However, I will want you to be familiar with the parts and the function of the parts of the microscope. Refer back to your Lab 1 submission where you took an image of your microscope and labeled it. Lab number 2 dealt with Microbiology Lab Safety. Again, this lab's focus was really about the levels of personal protective equipment, things like your gloves, your gowns, coats, and goggles. Also, make sure that you are familiar with the safety procedures and practices of the microbiology laboratory. What are some of the do's and don'ts when working with microorganisms? Also, make sure that you're familiar with things like the materials that we use, such as bleach, to disinfect not only your work area, but also to destroy used samples. Also, this particular lab focuses on the biosafety levels. And if you recall from the lab question quiz, we worked with biosafety level 1, which are the microorganisms that cause minimal to no impact to human health. These ranges go all the way up to the BSL-4, which contains the highest probability of life-threatening disease and require maximum containment to work with. I would also recommend, again, getting in and understanding the procedures for your lab. Take a few minutes to go through and understand the process that you used to set up the lab. That's why the written summaries of your weekly procedures in your lab submissions is really important, because I want you to really reinforce and understand the process that you use to set up the weekly labs. For instance, the organisms that we use in each lab are really important. This particular one on hand hygiene utilized baker's yeast, which is known as Saccharomyces cerevisiae. Also, make sure you're familiar with the types of media that are used in the lab. Oftentimes, we utilize nutrient agar, which is used to grow a wide range of different microorganisms. For instance, when we did our airborne contamination plate, we were looking for growing as many different organisms as possible on the surface of the plate. These agar surfaces are used to grow bacteria. From there, we went into lab 3, which dealt with culturing and aseptic technique. This particular lab walked through what media is and what media it can be used for, including the different forms of media, liquids, solids, and semi-solids, as well as the specific conditions to prepare agar. So it being liquefied at 100 degrees and solidifying at 42 degrees Celsius. I would also make sure that in terms of the types of different growth media, make sure you're familiar with things like selective versus differential media. What does that mean? Or chemically defined versus undefined. Why is aseptic technique so important when preparing microbial specimens? Again, aseptic technique is unpacked in the background, especially when we are preparing a streak plate and getting our agar ready where we are able to obtain isolated colonies known as pure colonies where we have only one type of microorganism growing. The streak plate technique is really a dilution to allow us to spread out a particular microbial sample on the surface of the agar plate. For this lab, again, make sure that you get in and you unpack your two labs and be familiar with the procedures, especially the types of stab tubes and agar plates that were used to grow microorganisms. From there, we did get into lab 4, which dealt with structure and microscopy. This really looked at the differences between prokaryotic and eukaryotic cells, with specifics around the structures of the cells, but also differences in the cell walls between a prokaryote and a eukaryote. Again, make sure you're familiar with information about the structure of the cells. Be sure that you can compare a eukaryote to a prokaryote in terms of their organization. With regards to this lab, this particular experiment had 3 different staining procedures. I can't emphasize enough. Make sure that you understand the reagents. So what did you use to perform a simple stain? Crystal violet being the key. What did you use to perform a negative stain? And what series of stains, knowing which one is the primary stain, which one's the decolorizer, which one is the counter stain, and which one is the mordant, and in which order you use those stains. Make sure that you're also familiar with what the end result is. Simple stain is going to stain all the cells one color to allow us to observe morphology or shape, as well as arrangement of cells. The negative staining, which uses nigrosin, is going to allow us to stain the background, giving us visual details of our cells, which are not stained by the nigrosin. And then the gram staining stains based on differences in the microbial cell wall. Gram positives stain differently from gram negatives because gram negatives have much less peptidoglycan in their cell walls. Peptidoglycan is the key agent that comprises the gram positive versus gram negative cell walls. Gram positive cells staining purple, gram negative cells staining pink. Also, when we talk about gram staining, make sure that you're familiar with the different microbial morphologies. That if you were to look at a picture, you could define what a bacillus is versus a coccus versus a spiral-shaped microorganism. From there, we then got into the Eukaryotic Microbiology Lab. And again, make sure that you're familiar with the differences in eukaryotic microorganisms, including some of the major groups like the fungi, the protozoans, the algae, and of course, the helminths or parasitic worms. As we get into this, make sure that you understand the context of fungi. We talk about things like the yeasts, the molds, and the mushrooms. Boldface terms are always important to know. Things like dimorphic fungi, because they can change the different pattern of growth that they exhibit based on the temperature that they're grown at. So please make sure that you review key information. Also, this was the lab that you were growing up the Saccharomyces cerevisiae, or baker's yeast, and this is the one where we grew the mold on the bread and apples. Make sure that you're familiar again with the reagents. Why did we use lemon juice as an acid? What was acetic acid used for? Why did we use sugar? So make sure that you understand why we're using those different ingredients. What are they allowing us to test for in the experiment? And then lastly, Lab 6, which is this week's lab, dealt with allowing us to look at different requirements for growth for a variety of microorganisms. We looked at especially the physical requirements for growth, such as temperature, but then we also looked at some of the various aerobic and anaerobic conditions. Make sure that you go through and you review the different types of aerobic and anaerobic growth, such as obligate aerobes, which require oxygen, obligate anaerobes, which do not grow on and are actually poisoned by oxygen. And then some of the other conditions, like microaerophiles, which grow at very minimal concentrations of atmospheric oxygen. Again, going through and understanding the procedures for the lab. We also looked at some different chemical germicides in which we grew microbes near discs, paper discs, that were soaked in various different chemical agents. Make sure you understand how you know and how you read the petri dishes after the microbes are grown near those discs. Also, make sure that you're familiar with the difference between why we would use a positive control versus a negative control. What's the reason why we would use positive versus negative? And especially in an experiment like this, what was our control? Again, we talk about having a control to make sure that our results are accurate. Think about, if there is an experiment, what's the control that we used in that experiment? So with that said, those are the 6 labs that comprise our first lab practical. And just some key tips as you go through and begin to review your labs on parts that you should emphasize as you begin to study. As always, lab office hours will be this week. Please come by if you have any questions about any of the content material. Best of luck studying, and thank you for your time.